Welcome to the NIH Distinguished Lecture Series. This is the last for this year. This series started in 2001 as an extension of the Falk and Rodler Memorial Lectures. And since then, it has featured a number of diverse, prominent scientists, nationally and international scientists, covering many of the disciplines that we are interested here at the Institute. And today is my great pleasure to introduce Phil Zamore. Phil is chair and professor of the RNA Therapeutics Institute at the University of Massachusetts Medical School. He got his PhD from Harvard University, working with Michael Green, investigating RNA splicing and splicing factors. And then he moved to the Whitehead Institute for his postdoc with Ruth Lichtman, where he was first exposed to small RNAs. And when he started his group at UMass, he made one after another great contribution to the field of small RNAs. One of her first paper of his lab was to define how double-strand RNAs are processed to generate the siRNAs. Then he contributed to the microRNAs. He found that enzyme that cuts the double-stranded RNA was also critical for the biogenesis of microRNA, bringing these two fields together. Then he also made many contributions, and more recently to the field of piRNA. He showed that the biogenesis is not dependent on dicer. They have a very special biogenesis. They are expressed from clusters, and the group of Phil was the first one to show which was the transcription factor driving the expression of this cluster. And more recently, he has been doing incredible work on the biochemistry and the enzymology of these piRNA enzymes, these PU enzymes that give us a lot of insight into the biology of these small RNAs that are expressed in the germline. So Phil received numerous awards and recognitions. Just to mention a few, he's been a Howard Hughes medical investigator for over a decade now. He's also a member of the National Academy of Medicine, a member of the National Academy of Sciences. And in addition to all this fundamental work on the biology of small RNAs, he's also an inventor. He's actually a fellow of the National Academy of Inventors. He owns several patents, and so he's translated all of these research. And he founded a now publicly traded biotech that developed one of the first FDA-approved RNA therapeutics. And this was in 2018, before RNA and therapeutics were put together in the same sentence. So really pioneering that. But today, Phil will talk us about the small RNA, their biochemistry, and their impact on germ cells and cell differentiation. So without further ado, I want to thank Phil, and we are looking forward to your lecture.
Thank you for that generous introduction. And it's been quite a while since my lab has worked on siRNAs because we've gotten very interested in what these small RNA pathways do in mammals. And although there are now 5 FDA-approved drugs that are siRNAs, outside of mice and rats, it's not clear that any mammal really has siRNAs. But fortunately, we still have an enzyme, Argonaute 2, that can use siRNAs and cleave a transcript. On the other hand, if you were a fruit fly, you'd be very grateful for your siRNAs because they would be your main adaptive immune defense against viruses. And that's true also of plants. And so what we've become very interested in is this pathway that's animal specific called the piRNA pathway. And what distinguishes piRNAs from siRNAs as Morgan alluded to, is that the biogenesis is different, but the endpoint is almost identical. So you begin with single-stranded RNA rather than double. So if I can, oh, there we go. But what I've discovered over the last post-pandemic period is that no matter what kind of science people do, they all know about CRISPR-Cas9. And there was a time, I remember, when people were hinting in the literature that CRISPR-Cas9 might be bacterial RNAi. And I find myself in the slightly unusual position now of having to explain piRNAs and RNAi in the context of CRISPR-Cas9. But it turns out it makes a very poignant comparison, because one is a bacterial protein that's only found in eubacteria and archaea. It only targets DNA, as best we know. There are other Cas proteins that target RNA, like Cas13, but Cas9 only targets DNA. It uses RNA guides, but the RNA guides, although we can program them with specific sequences, they must have a series of handles that allow them to be recognized by the Cas9 protein. So there's a constant portion. Here, there's actually 2 stem loops, and a part that we can vary that provides the specificity determinants. In contrast, the broad family of Argonaute proteins is found in both prokaryotes and eukaryotes. So archaea, eubacteria, plants, fungi, protists, and of course animals. It targets either RNA or DNA depending on the particular Argonaute. So for example, the Argonaute protein from Thermus thermophilus, which is a high temperature eubacterium, lives at 72 degrees, can cleave RNA or DNA in the test tube, although in vivo, its main function is actually to cleave DNA and resolve the products of replication. Argonaute proteins can use RNA or DNA guides, and as the piRNA pathway demonstrates, those can be derived from either single or double-stranded nucleic acid, depending on the pathway. And then finally, there is no special structure or sequence that's required for an Argonaute protein to enter into a complex with its guide. It simply has to be 5' monophosphorylated, and in some cases, double-stranded and the right length. And although we typically draw the products of the dicer pathway as having 2 nucleotide overhangs, all of the therapeutics actually have 1 blunt end. So it really is not very picky. And as you can imagine, this can lead to a loss of control in vivo. And we are beginning as a field to believe that there are examples of piRNA generating loci that have actually gone over to the dark side and become selfish. So, as I said earlier, there are prokaryotic and eukaryotic Argonaute proteins. In fact, worms have expanded that family beyond all imagination to encompass more than 25 functional Argonaute proteins that are specific only to nematodes. But among the other animals, besides worms, there is this special clade of pee-wee proteins that are essential for fertility. In worms, there are pee-wee proteins, but they actually activate a novel worm-specific pathway that does the business of silencing. And so these pee-wee proteins were first discovered by Hai-Fan Lin, who gave them the name of pee-wee, meaning P-element-induced wimpy testes, a very classical fly name. Imagine if you were a clinician having to describe a mutant in this pathway. And the pee-wee proteins therefore bind piRNAs, or Pee-wee interacting RNAs. So siRNAs are made by the enzyme dicer from double-stranded RNA, and they function through the Argonaute 2 protein in viral defense. Argonaute 2 in the viral defense pathway is not Argonaute 2 in people. There is an annoyance in the literature that everybody was discovering Argonaute proteins all at once and didn't coordinate the numbering. And so there is no Argonaute 2 that does viral defense in mammals. Instead, Argonaute 1 in flies is Argonaute 2 in mammals. And I apologize that, but fortunately, I'm not going to talk about Argonauts very much. The microRNA pathway, which is the dominant dicer-dependent pathway in animals, uses long RNA transcripts that contain a stem loop. It's then processed into just the stem loop in the nucleus by drosha, and then transported to the cytoplasm where it's cleaved by dicer and loaded into one of the 4 mammalian Argonauts. In contrast, piRNAs begin as long transcripts, and in animals that aren't mammals, are cleaved by piRNAs deposited maternally into the embryo to initiate the pathway by producing a 5' monophosphorylated end. And that end binds a Pee-wee protein. There is then a downstream cleavage, trimming, 2' methylation, and all the time this RNA is single-stranded, which means that the piRNA pathway has to do something to protect what is otherwise a vulnerable RNA, because it has the exact same 5' end, a 5' monophosphate, of RNA degradation products destined for degradation by XRN1. And that special protection mechanism is spatial localization within the cell. And we believe that the pathway works in specialized compartments that some have described as potentially condensates, and also on the outside of mitochondria. Okay, the net result, though, as you can see on the right, is that in all cases, you end up with some kind of Argonaute protein with a single-stranded guide whose base pairing specificity will tell it where to go and what to do. Okay. And the ancestral function of Argonaute proteins is to cleave RNA. So we believe, based on the conservation of key catalytic residues across evolution, that both the Ago family of Argonauts, like Ago 1, 2, 3, and 4 in mammals, and the Pee-wee family of Argonauts, all were originally endonucleases that had a catalytic triad that organized a magnesium ion to generate a hydroxide ion that catalyzed breaking the phosphodiester bond, leaving a 5' monophosphate. And that 5' monophosphate can either be a signal to produce more piRNAs from that transcript, or it can just be a dead end product that's destroyed. What's curious is that despite 900 million years of Pee-wee protein evolution and close to 3 billion years of Argonaute family evolution, every single Argonaute protein cuts its target in exactly the same place. And the way we describe that is we count from the 5' end of the guide to the 10th position, which we call guide position 10, and across from it is target position 10. And it's the phosphodiester bond that links target position 10 and T11 that is always cleaved by an Argonaute protein. This gives us a very useful signature in pairing imperfectly complementary piRNAs to their products, because the 5' end here has to end up corresponding to base T10. All right. There are 3 main sources of piRNAs in animals. The ancestral role of piRNAs was to silence transposable elements, both in the germline and the soma. Many arthropods retain that ancestral pathway. Some have lost the somatic arm and only silence their germline transposable elements using piRNAs and piWE proteins. Then, in some arthropods in the soma, for example, mosquitoes, piRNAs are used to silence viruses in addition to transposons. And in this case, these viral elements are actually inserted into the genome and bear a record of the species' exposure to viruses so that they are prepared for future infections with piRNAs to quell the infection. In mammals, there are 3 sources of piRNAs. In the fetal testis, piRNA silence transposons. In the postnatal spermatogonia, so these are the cells that are undergoing mitotic amplification, piRNAs are generated from 3' UTRs. We do not know what these do, if anything. We really have struggled as a field to come up with a strategy for testing them because hundreds, if not thousands, of mRNAs make small numbers of piRNAs downstream of their coding sequence in the 3' UTR. And we believe that these are initiated by ancient relics of transposable elements that are no longer active in the genome, but for which there are piRNAs still produced. And then finally, in the meiotic germ cells, beginning at the packaging stage of meiosis, there are long non-coding RNAs that are dramatically depleted of transposons that are turned into piRNAs, and whose function remains hotly debated, even though they were discovered in 2006. These so-called packaging piRNAs, because in mice they appear at the packaging, although in hamster they seem to appear at the zygote, are unique sequences. And so unlike all the other piRNAs that people can work on, these have the advantage of mapping to the genome once. It's one of the reasons why teasing out their function has been so difficult, because unlike transposon silencing piRNAs, they're not obviously antisense to any other transcript in the genome. And they map perfectly only to their own locus. But they give us a very unique tool to describe the general properties of the pathway. So if we wanted to understand how piRNAs target a given transcript by imperfect complementarity, we wouldn't be able to study it with transposons because we can't take any one piRNA and be certain of what its targets are. They include things that are the perfect complement of the piRNA, that are one mismatch to the piRNA, that are imperfectly complement with many mismatches. And we know from studying Argonaute proteins that such imperfect complementarity can often produce more active cleavage than even perfect complementarity. And so recently, my lab has embarked on a series of in vitro and in vivo experiments to tease out the rules that piWE proteins use for finding and cleaving their targets. And we've used the packaging piRNAs as a surrogate that allows us to do that in a way that can then be generalized to all of the piWE proteins in the mouse. This pathway is essential for fertility. And in fact, as infertile men have had their genomes sequenced in order to explain their infertility, nearly every important gene in the piRNA pathway has been identified as a cause of male infertility. Now, because I'm going to talk about a spermatogenic piRNA pathway in mice, I don't want you to go away thinking that females don't have piRNAs. It is simply that mice and rats don't have piRNAs. As far as we know, all other mammals, hamsters, humans, macaque, all have female piRNAs, and they do transposon silencing, we believe, but it has not been well studied, in part because the only model organism in which it can be studied easily in the lab is the hamster, which turns out to be very hard for genetic experiments because embryo injections have to be done in the dark. So I've stuck to spermatogenesis in mice, and I hope that I can convince you that at least for spermatogenesis, we now understand the rules that allow a piRNA to find its target and to cleave it. Okay. So a bit of background. Argonaute proteins don't need anything else to cleave a target. They need magnesium, a guide, an Argonaute protein, and a target. This is true for DNA-guided, DNA-cleaving Argonautes from bacteria. This is true for RNA-guided, RNA-cleaving Argonautes in plants and animals. But unexpectedly, we discovered 2 years ago that Pee-wee proteins are terrible endonucleases. And they're not terrible endonucleases because they're bad at that function in vivo. They're terrible endonucleases because they need to be activated by a second protein, GTSF. And so whenever I discuss experiments that involve a Pee-wee protein cleaving a target, it will be in the presence of GTSF, even if I forget to mention it. Second, I hope to convince you today that piRNAs, Pee-wee, and GTSF were likely found in the last common ancestor of all animals. And finally, they have unique targeting rules that make them particularly well suited for silencing transposons because they use just enough complementarity to be highly specific, and they're not too fussy beyond that so that they can be tolerant to mismatches in ways that other Argonaute proteins can't. Okay. So the story began with Shen Wu, who now has her own lab at the University of Geneva. And as a postdoc, Shen, working with Katie, a staff scientist in the lab, generated an invaluable resource that has really enabled all of the studies I'm going to present today. And that was 5 knockout mouse lines in which each individual source locus for pachytene piRNAs, of the top 5 producing piRNA loci out of 100, were deleted with a small deletion in the promoter that abolished transcription, thereby eliminating piRNA production from that locus. And since these piRNAs are generally uniquely mapping to the genome, we know exactly which piRNAs those mutations remove. And then Katie combined those mutations to make all the potential double, triple, and quadruple mutants. So we can remove large numbers of piRNAs at a single experiment and then ask what happened to potential targets. And the work I'm going to talk about today was largely done by Ildar Ganetinov, who recently, it says next week, but the truth is it was in August, became an assistant professor at New York University. Okay. So Ildar took advantage of a technique that we had pioneered in a collaboration with Will Greenleaf's lab at Stanford back in 2009 to study human AGO2. And this method, called cleave and seq, backformed from its abbreviation, CNS, allows us to measure the cleavage rates for individual targets using a specific piRNA. So historically, when people do cleavage experiments, they load an Argonaute protein with a single guide sequence. They use sequence affinity chromatography to purify just that one loaded species among all the potentially loaded proteins in the tube, and then they incubate it with a radioactive target, and they look at the cleavage products on a gel. Now, in this case, instead of looking at the products on a gel, we're looking at the products by high throughput sequencing. And so what Ildar did was he made complexes of 4 different piRNA sequences with 4 different proteins. So when he loaded those sequences into Argonaute 2, he used the first 21 nucleotides of the piRNA, so it would be siRNA length. When he loaded them into the mouse Pee-wee protein MeWe, they were 30 nucleotides long, because that's the length they are for MeWe in vivo. And for Mealy, whose guides in vivo are 26, he used the first 26 nucleotides. We don't know for EF, EF is a freshwater sponge peewee, what the in vivo guides are, but we used the 26 MERS for those as well. So he had 4 piRNAs by 4 different Argonaute proteins, which 16 different combinations. And then he designed a microarray of oligos that encompassed for each of the 4 piRNAs between 8 and 10,000 target variants, covering every 6 nucleotide contiguous stretch of complementarity up to complete complementarity in every possible register, and every single and double mismatch among those. And for all the singles, we were able to cover all 3 bases other than the complementary one. And for the double, the tandem mismatches, we turned them both into the most clashing bases possible. And then for each of these, he took 11 time points of reaction, and each reaction was then sequenced for the loss of the target and the gain of the cleavage products. And because reviewers are cruel, he did this with and without GTSF1, and ultimately in 3 independent trials. So if you wonder why I think Ildar is superhuman, we can do the math. So he did 4 piRNAs by 4 Argonaute proteins, by 10,000 targets, by 11 time points, by 3 replicates under 2 conditions. And the day before he left to start his own lab, he just popped in and said, I made 2,700 sequencing libraries in your lab. At which point I thought, and I paid for them. It's a bit of a shock. Okay, so we asked 4 questions. How does the length of complementarity affect the rate of cleavage by a piRNA directing a peewee protein versus an Argonaute protein? How do mismatches affect cleavage rates? How faithful is the selection of the sisyl phosphate? So are those rules that cleavage is always between T10 and T11 true no matter what the mismatch pattern is? And finally, are peewees actually different from Argonaute proteins? And if not, why do we have them? And if yes, why do we have them? Okay. So the design was very simple. The oligo was transcribed into an RNA that I've illustrated here, where the central portion was where we varied the sequence. And that allowed us both to look at the flanking context and all of the possible variants that I described for you. And then the constant regions were covered with blocking oligos so that we couldn't accidentally form a competing secondary structure between the constant region and the variable region. And then it was mixed with one of the 3 peewee proteins, or Argonaute 2 as a control, that had been loaded with a piRNA and GTSF1 and incubated from time points as short as 30 seconds and as long as 960 minutes. And we did the incubations at 33 degrees because that's actually the temperature, internal temperature of a mouse testis. Then if that particular piRNA can cut the target, it will get cleaved into 2 fragments. And by inserting a barcode here, we can clone and sequence this fragment and then know what was upstream in the original library. And so in addition to losing this target in one library and gaining this in the other, we can infer which complete sequence was present when we recovered this cleavage product. Okay. Then finally, we fit the data, which was a time course, to what's called the burst and steady state equation, pre-steady state phase, and then a steady state phase. And what we found was that mainly for the most complementary sequences, there was a burst and then it got stuck. And so we used the initial burst rate or pre-steady state rate to fit the data and compare it ultimately. Okay. So how does length complementarity affect cleavage rates? So here, for example, is an endogenous piRNA. In this case, it's actually not a pachytene piRNA, but the most abundant piRNA that silences the most abundant and active transposon in mouse, L1MC. And what I'm going to show you is a heat map in which red is very rapid cleavage and green and blue are very slow cleavage. Okay. Here's an example of complementarity beginning at base 7 and extending to base 25. And for those of you who have ever read anything about Argonaute proteins, you will immediately note that this is a very strange kind of complementarity, because if you know anything about microRNAs, for example, you know that there's this magic seed sequence from positions 2 to 8 that is absolutely required for binding. Okay? And so if we did this experiment with an Argonaute protein, for example, we wouldn't expect cleavage. But if we go to the heat map with base pairing starting at position 7 and ending at position 25, orange tells us that this is relatively efficient cleavage despite the fact that there's no seed match. We can look at MeWe and sponge peewee, and we essentially get the same rate, although MeWe is overall a more effective endonuclease. Both mealy and sponge peewee follow the same pattern of permitting blocks of complementary sequence that don't include the seed sequence, as well as blocks that do. And in fact, if you look here, this is complementarity in MeWe and in mealy that begins at base pair 11, no upstream complementarity, and extends all the way to the end, 30 nucleotides at the end of the guide. And we see cleavage, not just, perfectly fine cleavage. And the problem here is, of course, how conceptualizing base pairing that begins after the cleavage site, right, with no upstream cleavage. So how can you position the target in the protein under conditions like that? Then we looked at mismatches. Here's an example of fully complementary target. It essentially cleaved all of its substrate in the first 30 seconds of the reaction or so. And then here is a double mismatch in which positions G10 and G11 are mismatched. And although there is a large change in the rate of cleavage from 2 per minute to 0.025 per minute, if we compare this to the same exact mismatch pattern at AGO2, it falls below our limit of detection. So to the best of my knowledge, this is the first time our lab has ever seen this kind of mismatch support any cleavage at all. So it's already telling us that something is different about peewee proteins. And so just to summarize the data across all of the different pairing schemes, in this case, we're looking at complementarity across the first 20 bases with a single nucleotide mismatch. Argonaute is already slowing down substantially, 3 orders of magnitude or so, if we have a single mismatch at the cleavage site. MeWe is hardly affected, mealy is hardly affected at all. If we do double mismatches, if we do this with sponge peewee, we see the same thing, suggesting that these biochemical properties are conserved to 900 million years of evolution. And for double mismatches, where Argonaute can't cleave at all, MeWe and mealy can. Same for sponge. Okay. And then finally, if we look at how faithful the selection of sisal phosphate is, we can ask, how do these mismatches affect the positioning of the Argonaute and its guide, the peewee protein and its guide on the target? So the way we do this is we take our original set of target data and the proposed pairing schemes for each complete and mismatched target, and then we compare the identity of the 5' end in that barcoding experiment to the position we expect. Now, of course, in these kinds of experiments, you get nibbling back of the RNAs simply because no lab is free of RNases. And so Ildar spiked in a series of controls that allowed him to estimate the rate at which you lose those cleavage products in vitro due to things that have nothing to do with the piRNA pathway. And what you can see is that there's, in general, good preservation of the 5' end, but slight shortening by one nucleotide of each of the spike ends. And you see exactly that amount of shortening in the actual experiment. But the majority of all the cleavage events, whether you have a mismatch at position 2 or a mismatch at position 11, are in exactly the same position, the phosphodiester bond linking positions T10 and T11. Even if you have two mismatches, the same phosphodiester bond is getting cleaved, which is very surprising because for Argonaute protein, it's pairing those two nucleotides that actually puts the phosphodiester bond in the active site. Something different is happening here, and I won't be able to tell you what it is yet, but we are actively working to solve the structure of the complex of target, MeWe, and GTSF1. Okay. No matter what the contiguous pairing scheme is, even when pairing begins at position 11 and there is no upstream pairing at all, we see exactly the same phosphodiester bond getting cut. Okay. Then finally, is the real difference here GTSF1? Is it the fact that this protein needs a helper protein that gives it this promiscuous set of rules where if there's sufficient complementarity, let's say more than 16 nucleotides of contiguous complementarity, for each additional base pair, you can start unpairing other bases, even at the active site. And to analyze that, we plotted on the x-axis the relatively slow cleavage rate in the absence of GTSF1 and the fast cleavage rate in the presence for all of those L1MC targets from the cleave and seek reaction. Do you see for AGO2, red is a mismatch somewhere between T9 and 13, blue is fully matched, and all other pairing schemes are in gray. So for AGO2, GTSF1 has no effect. For MeWe, GTSF1 accelerates cleavage of all of those targets. And there's no pattern. Here's a target that is has one mismatch and is cleaved relatively slowly. It's accelerated to a similar extent as one up here that has a mismatch in a completely different place. The same is true for mealy. Mealy is not quite as good, so the acceleration is not quite as strong, but this is a 10-fold acceleration. This is a 100-fold. And even for sponge peewee, which is a relatively slow enzyme, sponge GTSF1 accelerates cleavage for all of them. So we don't think that the rules, the unusual rules for pairing that appeared to come out of our in vitro experiments have anything to do with GTSF1. So on the one hand, we have the unusual property that peewee needs a helper protein, and on the other, we have its strange rules for pairing permitting cleavage. Okay. So hopefully all of you are saying, that was an awful lot of biochemistry. What about the truth in vivo? And so, as I said, we have these mouse mutants that remove large numbers of piRNAs. And so what Ildar and his collaborators did was sequence 5' monophosphorylated RNA from purified mouse spermatocytes. So the 5' end of these has just one phosphate, the characteristic product of any Argonaute protein cleaving a transcript. Most of these sequences will be junk. About 85% of these are just mRNA fragments from ordinary decay pathways that we've caught in the act. Additionally, these products, if they don't re-enter the piRNA pathway, and most of them don't in mouse, are extremely unstable because they are the ideal substrate for XRN1. And unfortunately, our efforts to remove XRN1 have not helped. They've just made the animals sick. So in order to do these experiments, each single data point had to be 1 billion reads. And because we're sequencing so deeply to see such rare products, we end up with a stochastic sampling problem that I'll explain in a second. So the fundamental technique is called degradome sequencing. It was initially invented by Pam Green and Dave Bartel to sequence the cleaved products of microRNA-directed cleavage in plants. The 5' monophosphorylated RNA can be easily cloned by ligating an adapter on it. And then we match those up to all the piRNAs that have been sequenced and that whose abundance is sufficient that it should be present in every cell. So that's about 10 molecules per cell. And we look for reads that have at least 6 base pairs of complementarity in any register, and whose 5' end is predicted to lie between positions G10 and G11 on the piRNA we're proposing might have cut it. So for example, here's a cleavage product. We map it back to its transcript. We find a piRNA that complements this sequence so that the cleavage product is predicted to lie at T10 and T11. And that, therefore, is potentially explained by cleavage by this piRNA. And we restrict our analysis initially to piRNAs only from the 3 loci that we have combined into a single mouse for which we have a deletion. So we look at piRNAs that are unique to pi 2, 9, and 17, and we compare them to a mouse in which those piRNAs have been genetically removed. Okay? And we require that this cleavage product here decrease in abundance by at least 8-fold. In many cases, we just can't detect it anymore, but we require that it disappear by at least 8-fold. And that means we eliminate from the pool cleavage products that might clearly be explained by a piRNA, but were in wild type detected in 2 molecules per cell, and then in the mutant undetectable. So now we're requiring that we have a relatively high recovery of that cleavage product. Okay? And then we predict that if that piRNA really cleaved it, when we remove it genetically, the cleavage product should disappear and the target concentration should be unaffected. Okay. Oops, wrong way. Too many buttons on the mouse. So I want to point out that our analysis does not rely on the relative abundance of piRNA A versus piRNA B. We're only comparing A to A in different genotypes and B to B in different genotypes. Okay. So Ildar took that data and analyzed a series of what I would call sensible criteria that have been applied to other kinds of Argonaute proteins, and asked, what are the best predictors of whether a piRNA is likely to have made that cut, because in the absence of the piRNA, the cut product disappears. And he considered piRNA concentration, predicted binding energy, the number of paired nucleotides, the pairing of specific nucleotides, the first nucleotide identity, and the target site location. So if we were talking about a microRNA, for example, the concentration of the microRNA might be important, but not terribly important. Its predicted binding energy would be relatively unimportant. The number of paired nucleotides could be as few as 6, and they would have to be in the seed sequence. The T1 position would be most likely to be an A, and the target site location would most likely be in the 3' UTR. And so then he fed all of this data into a machine learning algorithm, and what he saw was that the most important attributes of, there it is, of piRNAs being productively cleaving an mRNA in vivo were first the concentration of the piRNA, which we used spikins to determine in molecules per cell, the predicted binding energy, or the surrogate for that, the number of paired nucleotides. What he didn't see was any strong contribution of the seed sequence. He saw a modest contribution of the seed sequence, and essentially no contribution, no requirement for pairing at any specific position across the piRNA. Nor did he see a T1 nucleotide bias or a bias toward being in a particular part of the transcript. So these rules are quite different from those we would get for Argonaute proteins and suggest that the Peewee pathway has a completely novel set of rules for what it can find as a target and cut. So that's either good or bad. So it could be bad if those rules allow it to cut our own transcripts, and it could be good if those rules allow it to suppress transposons escaping being cleaved by mutating their bases. Okay. So what Ildar did is he considered the rules for AGO2, in which a 21 nucleotide siRNA must be paired in the seed sequence, and in order to cleave, the siRNA must zipper up across the center of the siRNA target pairing in order to move the target into the active site and be cleaved. And pairing up to about position 18 continues to improve the efficiency of that process. In contrast, in the presence of GTSF1, mismatches are tolerated at any position as long as you have more than 16 base pairs, and you can have sites that include the seed sequence and sites in which the seed sequence is unpaired. So first he took all of the k-mers, in other words, words in nucleotides of different lengths from all of the transcripts in the cell of a mouse spermatocyte, which essentially covers almost every gene, and all of the introns for all of those genes, and asked, if we look at all of the transposons that are known to be active in the mouse, how likely are we to find the same word? So maybe a 3 nucleotide letter word, a k-mer of where k is 3, in both the exon or intron and in the transposon. And unsurprisingly, when sequences are short, there are lots of fortuitous matches between transposons and the exons and introns. So for example, you could have 12 nucleotides in a line element that also appear in an intron or an exon. But by the time you get to 15 or 16 bases of complete complementarity, there is essentially no way of making the piRNA more specific, because those k-mers are as almost as unlikely to be found as they would be if we lengthened them. That's exactly the solution that nature has come up with. And then if we ask the converse question, if it has to have 16 nucleotides of complementarity, but if it's longer, it can permit mismatches, is there any benefit in which the piRNA pathway would appear to be an advantage over the siRNA pathway, which silences transposons in plants? So Ildar simulated cleavage by either AGO2 bound to a 21 nucleotide siRNA that cleaves the most active transposon, L1MD, in mice. And at each generation in the computer, he mutated a single nucleotide, but any nucleotide that changed the coding potential of the genomic transcript of the transposon was excluded. So the rules that he used were there had to be fewer than 5 mismatches, there had to be fewer that or equal to one mismatch between G2 and G8 in the seed sequence, and no mismatches at G9, 10, 11, and 13. I think in the field, people would agree are the standard rules for functional siRNAs. And then the rules that I told you came out of his experiments in vitro and vivo were used for mealy with a 26 nucleotide piRNA, cleaving the exact same sequence. And after 1,000 generations, he asked, how many of those guide RNAs that occur naturally in vivo that target that active transposon consensus sequence no longer are predicted to be functional? So what he found was that the rate at which you accumulate resistance to cleavage using mealy versus AGO2 was fourfold different. So in other words, using piRNAs likely gives animals an advantage of keeping their transposons silent, both as mutations accumulate in the transposons and when new elements of related sequence invade the genome, suggesting that that's why this pathway became fixed in animal evolution. So I'll end there, and I will thank Katie Caccini and Ildar, who did the lion's share of the work, Amina, who discovered GTSF1's function, and Pei Xuan, who started all of our efforts to make piRNA mutant mice. And thank you for your attention.
Thank you, Phil, for that wonderful talk. So now questions. I'll ask the people in the audience to come to the mic and the people on Zoom to raise their hands or leave the comments in the Q&A. We'll start with Paul.
Lovely talk. I'm really curious about catalysis and how the enzyme can tolerate either a base paired cleavage site or non-base paired cleavage site.
Right. It's a great question. So there is currently no structure of PIWI with a piRNA bound to a target in the catalytically active conformation. We are trying to get the structure with GTSF1 bound, which should be in the active conformation, but we haven't yet had success. That said, if you ask AlphaFold what's going on, it suggests that there's a single glutamate that in AGO2 is pointing at the catalytic magnesium, positioning it for cleavage, and that in PIWI is facing the wrong direction, as if the rare time it swings in, GTSF1 might lock that conformation. The central channel where all of that catalysis occurs, even in the non-catalytic conformation of PIWI, is clearly much bigger than the central channel of AGO2. And so what we imagine is that for AGO2, base pairing across the cleavage site actually makes a rigid helix, which is energetically disfavored for binding, but forces the conformation to change sufficiently that the magnesium is very close to the target. Instead, in MIWI, our hypothesis is that we have a very accommodating large area where the target is essentially single-stranded, no matter whether it could base pair or not, when it gets cleaved. But that the problem is that it's not often in the right conformation to position the magnesium ion. And that's GTSF1's job. And I actually talked about some of the data yesterday at Duke for that. But I think right now we can exclude things like GTSF1 helps with target finding. We can exclude that GTSF1 helps with turnover. We probably can exclude that it helps the complex stay together over more time. And so by default, the only thing I can think of is that it actually captures a conformation.
Thank you.
You're welcome.
Phil, I have a question. So in your in vivo data, you show that for the activity of the enzymes, there was still some influence of the seed region.
Absolutely.
I wonder if you capture the same in the in vitro data, or there is something helping or modifying the activity in vivo.
No, we do capture that in the in vitro data, and it's subtle, but it's there. And I think there's a really good explanation for it. So if you look at the structure, and I'll try to do this with pantomime, but if you look at the structure of an Argonaute protein, the so-called seed sequence has the bases stacked in a perfect half helix. If any of you have ever used a program like PyMol, you know that you can click a button and turn one of the components off, and it literally looks like you turned the target off because it's such a perfect helix for the first 4 bases. And 4 is the magic number for nucleic acid hybridization. Organization of those 4 bases, like when you do a southern blot or PCR primer annealing, is what limits the rate at which a productive zippering occurs. And when your nucleic acids are naked, the bases are only occasionally aligned properly. So it means that the rate of productive collisions is 1,000-fold less than you predict from diffusion. But for Argonaute proteins, including MeWe, the rate is actually diffusion limited because those bases are displayed, which means that the concentration of MeWe doesn't matter that much in the reaction when they're seed pairing. But there are many piRNAs whose concentration in vivo is nuts, 100-fold above the KD. In aggregate, piRNAs are 6 micromolar in a spermatocyte, which is just shy of the concentration of ribosomes. And so what we think is happening is that, and we're testing it, but we haven't finished those experiments, is that when you don't have a seed match, you're now only getting cleavage from very abundant species because you need high concentration to drive on rates. And for seed matches, lower concentrations will be adequate for getting cleavage done in a reasonable time.
That was a really beautiful talk. So I have a question about your high throughput screen when you were looking at mismatches. Did you consider all types of mismatches? And in particular, what happens when you have GU wobbles?
So we considered all single mismatches. So there were 3 potential mismatches and all were represented. And GU wobbles are not permitted. So they act just like regular mismatches.
I have another question while we wait for others. Do you know what is the temporal activation or expression of GSFT1 in the elongation of the spermatids, and if there is any change in the cleavage between the round and the elongating,
Right.
that might drive, I don't know, decay of mRNAs like in a targeted manner?
So until recently, we hadn't sequenced cleavage products from elongating spermatids. So I can't yet tell you that. We are going back and doing it. What we see as you progress from primary spermatocytes to secondary spermatocytes to round spermatids is that the piRNAs and the piWe proteins come up because a transcription factor called AMIB gets turned on by STRA8 and myosin, which are the responders to retinoic acid. When that comes on, it turns up or turns on the source loci, MeWe for the first time, boosts the level of MeLe, GTSF1. And despite that, you see a delay before you get a change in steady state RNA levels. So you see cleavage starting in packaging spermatocytes. But by round spermatids, you see that those few targets of piRNAs, the steady state level is going to decline. That's where the biggest change is. In terms of GTSF1, there are 3 GTSF1 paralogs in mice. Before we started working on the protein, they had been deleted. So the GTSF1 mutant is completely infertile, and it is blocked at the onset of meiosis because it actually fails to silence its transposon. So it's required for mealy-based transposon silencing. At meiosis, this level goes up. And then the other two, GTSF1-like and GTSF2, were deleted in a double mutant and has no phenotype, which we don't understand, because in silk moth, there are two GTSF1 paralogs, and one is specifically able to activate cleavage by one of the two silk moth peewee proteins, and the other only does the second. So one is devoted to AGO3 and one is specific for seaweed. And we would have expected the same behavior. So we're not sure what we're looking at here.
Thank you very much. So being noon, I think we'll thank Phil once again for the wonderful talk.
Thank you.